Okay, so hopefully this works on the other end too. Yeah, so thanks a lot for getting up so early in the morning. So, it was 6 o'clock or so, no, 6:30, yeah. And Lisa has already been making fun of me because I think, I'm glad it's so warm here. I don't have to do this in my skiing gear, right? Because I'm actually no longer used to the weather, that I experienced as a kid. I experienced similar weather, but in California you become so mellow. Yeah, it's kind of over.
You look cold. You really cold.
Yeah. So I'm gonna first of all point out that it takes a village to do these things, and I'm the most useless person in this. All I do is wear pom-poms and try to cheer these people on. Yeah. And without their help, none of this would actually work. And what we're interested in the lab mainly is understanding how sort of quantitative ideas from physics can be applied to biological systems, and how we can then maybe make some sense in some predictive way about how embryos are getting in shape. And we've seen great examples, like Stefano yesterday rocked the show with how phenomenology can actually be brought in there. And so the way that we study this is essentially using live imaging. So we take similar ideas as what Sean has shown you yesterday, this in toto microscopy. You can see here a whole zebrafish embryo recorded by postdoc Susie Wopat, who has established all this zebrafish stuff in the lab. And when you're looking at the fish, you could kind of feel that there's an overwhelming complexity in the sense that there's 10,000 protein coding genes per cell, and there's about 10,000 cells, right? So this can be easily overwhelming. But now looking at the video, what you actually observe is that this is very well coordinated across scales, right? And you can see that this is a very robust process. In fact, once you've seen one fish, you've seen basically all of these fish, right? They all will do the same thing. And so, the hope is that you can actually get there and do some form of quantitative predictions, if you like, yeah? And lots of focus, that has been spent over the past decades was to figure out how genetics is important in these processes, right? And a very, very striking example that underscores this really strikingly are the Hox genes. Yeah? So you mutate one single gene, and instead of a pair of antennae out of the face of this fly, you now have a pair of legs growing out of the face of this fly. Yeah? And this is a true remarkable success for biochemistry, right? We know actually where we need to make a point mutation in order to change the body plan of the animal in radical kinds of ways. But at the same time, we're at a huge miss, right? Because we know neither how you make an antenna nor you make a leg, right? And we can study the biochemistry up and down, and we still didn't really understand how that actually goes. But there are some useful sort of ideas that are coming in from this, and that is, for example, what many people have kind of understood as a metaphor, right? So where you say, okay, you have this famous Waddington landscape, you have cells that can differentiate, and they're rolling down, this landscape. And I think Paul-François has shown a talk earlier that you can actually take this metaphor to something more concrete, right? Where you can ask, how does this landscape actually direct, these fate decisions, right? And fate is still very much the center of the attention for many people. And so, for example, here is a truly remarkable success. I find I was totally flashed when I saw this the first time. This is a synthetic mouse embryo. I think that's what they call it. I would call it a reconstituted mouse embryo. What you do is you take 3 different types of stem cells, you throw them together in some well-defined kind of condition, you grow these things up, out comes some structure like this. Yeah? So this is in mid-gestation analog to a mouse. Yeah? So 8 days within, the pregnancy. And what I find remarkable about this thing is that its geometry looks kind of like that of a mouse, and there's gene expression patterns that are kind of similar, right? So indicating that actually you can trigger morphogenesis without mom and dad, right? But what you have to bear in mind is that this is actually a relatively rare example. Yeah? When you're going to earlier times, you can see how, in this dish, there is many, many structures of different types of shapes emerging. Yeah? And only a handful is actually looking like the mouse embryo at 4 days should look like, this characteristic kind of cup shape, right? And you can control the biochemistry really well in these settings, right? Of course, we can think about variability potentially being there, but there must be more missing because these shapes are really, really different, right? And so one thing that we have been arguing is that this is essentially involving mechanical signals and geometric, components that are important, and that there's going to be some sort of a feedback, between signaling mechanics and geometry that is going to trigger, development itself, right? And so what I'm going to do is I'm going to talk a little bit about how maybe we could extend this Waddington landscape picture, which happens in gene expression space mainly, to now the physical space, right? So the physical space being, mechanics, geometry, and gene expression together. So this is really high-dimensional space, and what I'm going to, hopefully get across is that we can think of morphogenesis as a attractor manifold. So it's some kind of a manifold, that, attracts the embryo towards this, right? And then the embryo is going to develop within this manifold. So, this is like the marble rolling in Waddington's landscape, but now, using these physics extensions to it. Yeah? And what you can then think about is that, there's developmental dynamics, like the fly embryo, for example, that I like to study, will unfold as one line. Yeah? And all embryos will follow more or less that line. Right? And so when you're thinking about the space being very high dimensional, there's a lot of space that is not embryo. Yeah? And so I would like to understand the texture of this not embryo too. Yeah? And so depending on how this goes, I'm going to talk first about what we can do inside the embryo to learn, how this landscape could look like. And then I will move into some new direction where, we move out of the embryo and that we will then try to coin something like structured active matter, where we would like to essentially establish form and microscopic structuring, just like, you have in regular matter, but this time with activity. Yeah? Okay, so I'm going to quickly introduce you to the hydrogen atom of developmental biology. Yeah? I hope that you're not offended by that. Yeah? Maybe the C star is easier, yeah? But so basically, this is Drosophila. Yeah? So Drosophila is very good because you have lots of genetics, and it has this very simple monolayer kind of structure on an ellipsoid, right? And it undergoes very canonical kind of motion patterns that you also see unfolding in more complicated things like the zebrafish. So for example, you have axis elongation, and this axis elongation unfolds in this cyan tissue here, which I call the germ band here, and this is a common name for it, which folds over to the posterior side. And in this particular part, I'm going to share with you 3 little vignettes, essentially. One is the kinematics of these embryos is actually strikingly simple. Given the potential for complexity, they do something very, very simple. And then I'm going to discuss a little bit, the role of geometry and mechanics in guiding the dynamics of this process. And then hopefully I will be able to make some connection to, molecular level regulation of this. Yeah? And I guess, most of you have seen some of this textbook material for the Drosophila embryo. So Drosophila embryo is kind of governed with stripes that set up some sort of a coordinate system. And famously, the pair rule genes are actually being associated with playing a very important role in this axis elongation in Drosophila. So when you mutate even skipped, that's this green line that you can see here, you can see how the extension of the germ band, which roughly doubles over the course of like 40 minutes or so, is reduced strongly. Yeah, it's reduced, by 40% or so. So it has a quite striking impact. And the genetic perspective that comes into this is like a feed-forward perspective. Yeah? What you say is, we are encoding, an anisotropy in this embryo through the juxtaposition of pair rule genes. So when you're looking at the cellular level, you have a cell here that has high levels of even skipped, and its neighboring cell has low levels of even skipped. And so what you're going to do is you're going to recruit myosin to this junction. What that is going to do is going to shrink this junction, and then you undergo this T1 transition. You have swapped neighbors, and now you have microscopically extended this. And because the pair rule genes are organized over this large range, you coordinate all of these T1 transitions, right? And then you should get germ band extension, right? And, this raises lots of questions. One of the questions is, how do you go from a nucleus to the membrane, right? And there was some very strong contributions from Jen Salem's lab, where they found that there are receptors on the surface of each cell. These are toll-like receptors, right? But what they learned is that these toll receptors are actually isotropic, and they're not anisotropic. What that means is you need to have some other mechanism that causes anisotropy. So maybe there's some toll code or so, right? And you can go now for a hunt of how these interactors are actually looking like. But really the link, the molecular smoking gun that you would like to have is actually not yet there. Yeah. So there's something missing. Yes.
Maybe can you clarify what you mean by a receptor being isotropic?
Yeah, so the receptor is essentially distributed along the surface of the cell. here, right, at the apical side, and that is isotropic. Yeah, so and myosin's distribution is very clearly anisotropic. Yeah, and so how you go from that isotropic to the anisotropic signal is a non-trivial question. You had a question.
Is this the difference between the two types of cells?
That is the idea of this model, yeah, that at this point you end up getting this recruitment because this cell is one type and this is another type, right? And there is this anisotropy that is coming in. Yeah? That is the question. Yeah, so that is essentially what we don't know yet. Yeah, we don't understand this yet. We have ideas, that essentially these toll-like receptors could play a role in how they are making a code, right? So toll receptors are actually coming from immunity, and so they can actually be used to sort of differentiate there. Yeah? And so this would be the idea here as well. But how you direct this anisotropy with this is very difficult to understand. There are lots of arguments that one can go into detail, but I would fill the whole day here. Yeah.
Not going to got something to do with parallel genes or segment polarity.
That is the idea, yeah. So the idea essentially is that the parallel genes are essentially having a phenotype, right? But what you could say now is that most of this work that I'm presenting you here is essentially based on fixed samples. So what you have is endpoint assays. And what that means is that you've actually lost the fossil record of how you got there. And if I say that this is a feed-forward machinery, all of this works, right? But if I say it's a dynamic system, I'm beginning to struggle because I no longer necessarily know whether that is working or not. Does that make sense? Great. So, we've started to look at this now from a different perspective, and we came in from this kind of phenomenology through mechanics, right? So, with Boris and Nick in my postdoc time, we, imaged live these embryos with myosin markers, right? And you can see how this myosin is very nicely anisotropic along the surface, right? And you can feed this into, fluid mechanics, essentially, and you can say that, there are forces that are proportional to the anisotropy and the intensity of this myosin signal. And when you solve this equation, you end up getting a predicted flow that looks very similar to the actually measured flow. And the key feature that gives rise to this flow is that you have a dorsal ventral gradation of the myosin signal. No? And so, the gene expression tells us the AP patterning is the important thing, but the mechanics says it's the orthogonal axis, it's the dorsoventral actually that is kind of important, right? And this model works kind of well, not only in our system, but there are also other systems. I think Mattia might have spoken a little bit about, avian systems, right? Chicken. Francis Corson has really great work with the quail, very similar kinds of models. And I mean, Maha here has also worked with Mattia on these things, right? So you see similar equations work in many different systems, and this encourages that this approach might have something to it. But there are, of course, also issues with it, right? So myosin is not a free-floating kind of structure, right? And all I measure here is myosin. Yeah? But there has to also be some other stuff, right? So the substrate for myosin is actually made at the adherence junction, right? At these adherence junctions, what you have is cadherin dimers that are making the cells kind of attached to each other. Out of these come, actin filaments, right? And then the myosin interacts with these actin filaments. It sits on these actin filaments, and what it then does is it contracts essentially those cell edges, right? And this picture doesn't say anything about the connection to cadherins or actins for that matter, and that's what I'm going to try to get into. If I want to make such a connection, what I need to do is I actually need to establish many different markers imaged, at the same time, and that's unfortunately very difficult. To do this in the same embryo, you can't measure more than 2, I guess, at a time. So what we've decided to do is to compile essentially the data from the lab into something that we call a morphodynamic atlas. So what this is doing is it takes embryos, movies of embryos, and it time aligns them and aligns them in space so that they are kind of in the same developmental stage and that they're in some standard orientation. And what you can do with this now is you can look at how your embryo is undergoing kinematics, like how these cells are moving. You can at the same time visualize how the cytoskeleton looks like in this embryo, and you can also look like how the parallel genes are developing, right? And in this way, you get a relatively rich data set, which is actually publicly available for download for the theory friends who would like to work with this. Yeah? And you can do things like this, where you take the embryo and you segment the embryo, you track the cells, and then you can essentially visualize, how individual cells are contributing to this process.
through this, do you think you're getting, like, in the end, you get one data set that is sort of the average result line?
Yeah, exactly. So I think of this as an ensemble average, like, how physicists would think about this, I guess. Yeah. And of course, there are small differences between the individual embryos, but most of my work is happening at a coarse-grained level, yeah, where, I sort of audaciously forget about cells and try to understand the phenomenon of what happens in a tissue patch, right? Because an individual cell is not going to change the shape. It's the tissue patch that changes the shape, right? And so at that level, I can do the physics, right? So, okay, so when you have such an atlas, the first thing that you want to do is kind of characterize a little bit the kinematics that is going on in this embryo. And what you can see here is a simple autocorrelation map. So what we do is we take the flow field from the embryo at one time point and we compare it to, the same embryo's flow field at some other time point. And what you can do there is get a correlation, right? So a number that is 1 when it's red and -1 when it's blue. And so the diagonal of this is all going to be 1s because you compare one time to itself, right? So this is the exact same flow field, has to be 1, right? But what you're observing now is that during different times, you see these remarkable structures, these blocks, right, where correlation seems to be really, really high, the autocorrelation within this embryo over hours, right? And you can actually give names to these phases. So, yeah. Yes, averaging over the total embryo, total flow field. Yeah, everything I do is in total. Yeah. So, this essentially, this block here is germ band extension. This block here is germ band retraction. What is really nice is germ band extension anti-correlates with germ band retraction. Makes sense because germ band moves this way and then moves that way. Yeah. And you can do this, of course, also with 2 embryos. You've time aligned them, you shape aligned them. And what we then see when we're now zooming into this corner, because we don't want to record embryos and embryos and embryos for 18 hours, you can see that there's actually rich patterns, right? So, the cross correlation this time for germ band extension is basically 1 throughout germ band extension. And then there's this phase that is known as ventral furrow formation, where the ventral furrow internalizes, right? Where they also correlate extremely well in this time. And also during cellularization, which you can hardly see in this, there's a strong correlation, meaning these things are very, very well regulated, right? When you see one, you have seen them all. That's what this says, yeah? And what you can do now is you can do this ensemble average, right? You were asking about this, yeah? So you can take all these aligned embryos and you can define, an ensemble average, which takes every of these embryos essentially and says this is the typical germ band extension movie of time. Yes, please.
In the previous picture.
Yeah, I'm going there. I'm trying to go there. Yes, there.
Like between 4 and 8, there are stripes, blue and orange stripes.
Yeah. Yeah, very interesting. So what happens between these times is that the motion in the embryo is extremely small. And you have lots of cell divisions and stuff like that, right? So I think of these as maturation phases between these dynamics. Yeah? We're doing something similar in the chicken now, not in the chicken, in the fish. Sorry, oh God, these animals. In the fish, yeah. And what you see there is that there are no such maturation phases. Yeah, so they seem to have also such blocks, and they execute them right next to each other. Yeah, so animals can do lots of things, and the fly has phases where it's stationary or standing still, and then phases where the flow is going.
This is a fly feature that it correlates so well.
No, I see this in fish.
You do?
We see this in fish. Yeah, I'll have Susie talk about this. Yeah, so this is more her territory. But there's a preprint out there from Susie which essentially separates the germ layers in the fish and then does this kinematics within the germ layers. And the autocorrelation has these phases there too. And what we're working on right now is cross correlations. But you can clearly see these features. Okay, so we make an ensemble average, right? So we average many movies. In this case, this is 57 movies into one standard embryo, right? And what we can also do is we can time average the flow of germ band extension within each of these embryos, right? And then we can compare how is the time average of each of these embryos comparing to the time in the ensemble average, right? And what you can see is that this correlation is basically hovering right around 1, yeah? So, within what our error is, right? And what that means is that the ensemble average, in very good approximation, is the time average. Yeah? And that's remarkable because what this tells me is that the embryo actually has a stationary flow. Yeah, and that is sort of the simplest physics I could actually make happen if I want to get in shape.
This time average you're saying is within these modules.
Exactly. Yeah, it's within one module. Yeah, if I made the time average of the whole embryo, it would probably look like nothing. Yeah. Yeah.
And have you looked? So yes, most of it is basically extremely well correlated.
Yeah.
Where is the largest variation then?
Ah, that's a great question. Yeah, we haven't really looked at it, but I think that's a good question. And I think I want to translate this into the fish and look at the shape, because the fish's shape is interesting. Yeah, so in the fish there's lots of interesting features like an organizer and stuff like that, right? And maybe there are regions where variation is suppressed a lot. Yeah.
Do you look at the edges of these blocks and how the transition to non-correlation?
Yes, we've done something like this, yeah. And so how the transition to the non-correlation occurs, I took it out, I could show you the slides, is essentially germ band extension happens to encode a geometric feature, yeah? And this is how long the germ band becomes. And what we can do is we can manipulate, how fast the germ band essentially goes through temperature manipulations and stuff like that. And what we see is that the length of the germ band is what really matters. Yeah? And so you can actually collapse the speeds, the average speeds, onto some typical curve if you, correct for maximum velocity during the individual embryo, right? And that what that is, the product of time and velocity is length. So this thing encodes geometry. Yeah? So I would say what is happening in the fly embryo is that you want to achieve a certain geometry, which is this fully extended germ band, and then you stop doing this. How and why and all this is an interesting question. And there are some theories out there where, Fridtjof Brauns and Nikolaus Clausen have essentially proposed that the initial state of the tissue, the degree of ordering within this polygonal tiling actually encodes how long you become. But that's something that still needs to be tested.
Is that information that propagates from one point, or is recorded everywhere at once? That makes sense.
I guess it would be recorded everywhere at once because it's the ordering of the lattice, right? And so this is like a macro property. Yeah.
How early development can this registration and alignment be reliably applied?
I would imagine that even in early times, like, the early cell divisions, that you see in the embryo, you could do this because they are having been recorded as very, very precise, right? So what was it, 8 minutes, Stefano, I think, right?
I mean, they're very precise, but they're not always start in the same place, but.
Yeah, but the timing of these things can be done really precise.
Yeah, wave goes through them in a couple of minutes.
Yeah. So I think this could almost be going all the way to the first, but we're looking more at the cellularization time. That's when we start looking at these processes. Yes.
And maybe you said this, to be honest, I was distracted for a couple of minutes, but if the genes control opposition myosin and myosin controls the flows, how do you get a stationary flow?
Great, Stefano, thank you.
So is there a time where they set the flow and then everything keeps going?
Okay, so I'm going to say an opinion. It's a little unpopular. I'm going to say this is a great idea that parallel gene juxtaposition is important for myosin, but it's an idea. There is zero proof out there that it is true. Right? There's one paper that shows juxtaposition could be important, and that's it. Right? And they are not looking at time courses. They're only looking at endpoint assays. And I can get in many different ways to the same endpoint, right? And so it's great work. It helped us a lot to understand and think about this, right? But I actually think unless you show me a video that shows these two things at the same time happen, I'm having a hard time to believe that this has to be the mechanism, right? That's where I am at.
Myosin is not anisotropically distributed.
Myosin is anisotropically distributed.
No, it's like the parallel gene mutants. It's lower levels probably.
Yeah, it's lower levels and it has an impact on this. I'm not saying that, the parallel genes play no role. What I'm saying is the anisotropy is not endowed by the parallel genes. Yeah. Okay. So what you see here is germ band extension, right? And what you see is that these parallel genes are sitting in a frame which is known as the Lagrangian system. They flow with the tissue, right? And what I've shown you is the tissue flow is stationary. It's not in the Lagrangian frame. It's actually Eulerian, right? And I'm not smart enough to understand how you go from Lagrangian control to Eulerian flow. Maha might have ideas, yeah, and that would be great. But, I'm not smart enough for this. Yeah? So I try to think the following way. The embryo does simple things, right? And so this is no longer simple to me, right? That's how I think about it.
The myosin part is stationary too.
Great, you've already done the talk, so it's stationary. Yeah. So essentially, what we have to do now is we have to go into what's the role of the geometry and the mechanics in setting up the tissue dynamics, right? And tissue dynamics is essentially myosin, right? That's what we are claiming, right? And so essentially, Stefano has sort of sold it already. Yeah. So I can go through this very, very fast. You have a fruit fly embryo, right? And this is what your typical flow field looks like. It has rich features, right? It has domains where these saddles are, right? That's where you kind of go straight down. And this orange domain is also the domain where most of the imaging happens, because it's very convenient, You can see this well, the cells stay within this domain, right, when you do a confocal video. Whereas when you're over here in this domain where the vortex is, the cells very quickly leave this domain, right? And so, what happens is here the cells are actually always moving parallel to the parallel gene stripes, whereas in run stripe 5-6, what happens is these cells are actually rotating. Yeah, they are rotating a lot. Yeah? And, Nicolas and Matt together, analyzed carefully how the myosin looks like. And what they are learning is essentially that the sourcing of myosin follows the dorsoventral axis. Yeah? It seems to be stationarily sourced. Yeah? And myosin is not living forever on the junction. It actually falls off within 5 minutes. Yeah? And why I'm emphasizing this here is because it's such cute work from Dina and Todd, in Denver, where they essentially showed, last year it came out on JCB, that you can have cells, that are basically, neighbors in even skipped, right? And they're in this region where this rotation occurs, and they're flipping into the DV axis, and all of a sudden these even skipped rich cells that are supposed to not be juxtaposed actually recruit myosin. Yeah? You can see here the time course. Unfortunately, the colors are a little different, but that's their paper, so I have to live with that. They measure the alignment of the junction with the DV axis. They see it comes in from much higher values, and they want to zoom on to this time when it actually flipped. And they see this very rapid flip goes along with very rapid recruitment of myosin. Yeah? So what this is interpreted in their paper as is that the junction moves into the DV axis and then it can recruit myosin, and so that there's an additional effect that essentially recruits myosin. Yeah? Jen Zahlen has actually also reported something similar like that in the Zegar paper where they look at, and it is always kind of interpreted as an exotic kind of phenomenon that the cells, in addition to parallel gene juxtaposition, also get this other thing, right? And what I'm trying to say is maybe we can make the worldview a little bit simpler and assume that it's just, the DV axis that is important. Yes, please.
Are there any signals in the chorion in the eggshell?
Yes.
That might.
Yes, you have it. You've understood it. Yeah. So wait for 10 minutes and then we're there. Yeah, you got the problem. So essentially, I'm going to try to set you up towards what Maha is now getting to from a physics point of view. Yeah. So essentially, what we have done together with Boris and Nick a time ago was come up with this active tension network that is now, I guess, kind of getting more popular. Yeah. So you can think of your cell.
Just sorry, before you get into that, can you just give a 1-sentence description of why physicists are excited by stationary flows? Like, I don't know.
Why am I excited by stationary flow? I could flow in so many ways to get into the final shape, right? I mean, there's tons and tons of ways, right? And if I have a stationary flow, then this is kind of the simplest possible way how my simple mind can think about, how you make the shape of joint band extension occur. Yeah? You always do the same kind of motion pattern, but your cells move through this thing. Right? And so if cells move and make the decisions, it's really complicated because what I need to do is every cell needs to get a thing of information, right? Where to go at that time, right? This is crazy. And when I now say, okay, I encode once this thing, right? I just have one-time information and then everybody follows this rule all the time, yeah? So it's simple, right? And
It's the simplicity then.
it's simple. Yeah, it's the simplest way how I can actually get there. Yeah.
So I'm just trying to think for something like germ band extension, how this is possible if I have the tissue, like, because I'm envisioning like initially I have this tissue up here, right, which is not the germ band. And then I'm moving around. You say, that, well, the flow is stationary the whole time. But like somehow there's a disconnect in my head that like these cells at the end of that, or, even intermediate in that flow, I start to have a pocket of cells up here on the anterior side that are moving that way, and they weren't.
So,
and so like,
so
I'm not seeing how
what you have is this amnioserosa tissue, for example, right? That kind of can compress. This is this dorsal most tissue, yeah, that can compress and stretch in the other direction, and that is what is occurring when you look at those cell shapes on the dorsal pole. You can actually see that they make major changes. And then another thing that I should say is that these are all the in-plane plane flow components, right? So this is essentially what happens in the plane of the tissue. And there is to some degree also out of plane stuff, right? Which I'm ignoring in this analysis. Yeah? So, this out of plane stuff will play some role. Yeah? And I'm not saying that I understand it 100%. Our predictions go to 90%. Yeah? But I think 90% is good enough with me because biology has all this enormous variability, right? So that those 10% is a lot of work to actually get there and understand. Does that answer your question?
Yeah, yes, and I'll talk to you about what I don't actually.
Yeah. Okay. Great.
Yeah.
So essentially, long story short, what Boris and Nick predicted is that there should be some form of mechanical feedback that equilibrizes a tissue. Yeah. So tissues are elastic. Yeah. On short time scales, come with laser cut it, and then you see that there is recoil, right? And that means tissue will be elastic. Yeah? And what you can say is that it's like a muscle, right? You have a walking kernel, right? And when there is too much load on the muscle, then the motors will start slipping. Yeah. And when there's too little load, the motors will start contracting, right? And so when I now look at my junction here, I have some motor on it, right? And I'm pulling on this junction, then, I'm going to be out of balance, right? So there's more forces on those motors than they can bring up with their stall force. What I can do is I can recruit motors, right? And when I recruit those motors, I'm getting back into force balance. And when you do this in one of these active tension networks, what you will say is that you should get myosin rate being proportional to the rate of strain. So this is very clear that how fast you recruit motors depends on how fast you're pulling on the junction. Yeah? And Hannah has done this, heroically in her PhD. She took optogenetics and all sorts of measurements of the embryo in many different ways. And what she finds is this really clear correlation between myosin rate and strain rate within these embryos, right? So, and this is satisfying to me because this was predicted before we did these things. Yeah, so this is kind of cool. Yeah, and what is also cool is that Pierre-François Len's group has done some orthogonal kind of measurements there. They took optical traps and they're pulling on junctions, shrinking these junctions, and they can show you that also in the other direction, of the branch, this feedback actually exists, right? And so this is really nice, right? There's some harmony between those two. And so the question that you can now ask is, how is this coefficient, which, is responsible for recruiting a good amount of myosin in the embryo, patterned over the embryo, right? So, there's positional dependence. You could be in the germ band, out of the germ band, could be different, right? But then there's also orientational dependence, right? You could imagine that it depends on some anisotropy that you have inherited from, some other system. And so Hannah sort of teased these things apart, and what she saw is that when you're looking in ventral regions, you get a very high feedback coefficient, which means in these regions, that are sort of here in this domain, you are very effectively translating mechanical feedback, whereas when you're in these dorsal regions, you get it actually weaker, and there's no dependence on the directionality of the junction, meaning that this mechanical feedback that we're measuring is actually isotropic across the surface of the embryo, and there are anisotropic mechanical insults that are changing it, right? So one of these is, for example, the internalizing ventral furrow that kind of pulls on these cells a lot, right? And that can lead to signals coming in. And now I'm going to Maha's question, how do you molecularly regulate this? Is there some factor, in the thing in the surface of the embryo? So what you can imagine now is we have our myosin junction, right? And we still talk about this substrate. And one question you have to ask yourself is, how do you measure strain rate as a cell, right? So because that's apparently what leads to myosin rate, right? And I would say this has nothing to do with the length of the junction, because strain rate is really some frequency, right, if you like. Yeah? And so, it's more like the density of something on the junction, right? And so we could postulate that there should be some sort of a sensor, right? And if I pull fast enough compared to the time scale of the turnover of the sensor on the junction, then I'll change the density of this thing. And that is what could trickle down towards, sensing of mechanical feedback. And so, now comes the tie-in. Yeah? So essentially, this is ecadherine in an embryo. It's something that sits on the adherence junction, and it's actually symmetry broken along the dorsoventral axis. It's very high on the dorsal pole and low on the ventral side. Mechanical feedback is also symmetry broken in that kind of way. It is low on the dorsal side and high on the ventral sides, makes it an interesting candidate. And Adam Martin and Ned Clark have shown that this is under control of roughly DPP signaling. Yeah? So BMP. Yeah, and BMP is actually sitting in the shell. Yeah, so that is where we're at. So what we did is we teamed up together with Vincenzo Vitelli and Jonathan Colin, and through movies, right, like we have from our atlas, into machine learning methods. And what this thing spits back is a relatively simple kind of model, where you say, okay, you have myosin sitting inside your cell, so it's advected with the cell. Myosin can detach, right? So it can fall off, right? We see this all the time in biophysical experiments. There's positive tension feedback. Tension creates more tension. And there's some geometric stress term, which comes from the hoop stress of the egg. And there's some strain rate recruitment. And what I want to point you towards is the red stuff. This is, cadherin here, which is symmetry broken along the dorsoventral axis. Interesting thing is you can replace cadherin with other things that are broken along the DV axis, and the same model comes out with slightly different parameters. So this makes it encouraging that essentially, this is a feature, that the dorsoventral axis symmetry breaking is important. And what you can do with this is you can actually do 30 minutes of germ band extension and myosin dynamics with this model. So you can see the time course of these things. And you can do this in mutants. And so, here's a twist mutant. You can do this in tolerant9 mutants and others.
All the difference between active tension and strain rate recruitment.
Yeah, so strain rate recruitment really cares about how you're recruiting myosins in response to a quick deformation, and it actually will lead you to equilibrium. You will stand still. And what this active tension is doing is it's saying if there is a junction that is singled out in some form by tension, it will actually recruit more motors so that it starts collapsing. So in some sense, active tension is what drives the process, right? And what the strain rate recruitment is doing is it makes sure that it is in equilibrium. And so we have this adiabatic kind of picture, right? Where you go from one equilibrium point to a new equilibrium point, and this new equilibrium point is determined mainly by active tension and maybe also this geometric stress. There's a really nice paper from Fritjof Browns that I can send you if you're interested in reading about these things. Yeah.
Can you explain the expression for the geometric stress? Like, does geometry?
Oh, this one? Well, it's hoop stress. It's basically hoop stress. Yeah, it's barbecue. Yeah, so you take your sausage, you put it on the barbecue, you heat it up, the pressure in the sausage goes up, right? And then around the circumference of the thing, you have higher stress than along the long axis, right? And when you do this long enough, your sausage will pop, right? And it will pop always along the long axis, right? And that's because the hoop stress is larger. Yeah, and that's what this is essentially.
So gamma is the tension. Gamma is the tension.
Yeah, that is adding to a bias in the tension essentially. Yeah, that's what this is doing. Yeah.
Are you saying that basically the only thing that really matters is the shape of the egg in the end? Or?
No, I think that there are two things. The shape of the egg is an important player, but there's also the symmetry breaking through the C field here, which is symmetry broken along the dorsoventral axis. Yeah.
So the bias and the feedback as well. So it's basically packaged in the bias and cadherin.
Correct. Yeah. So basically, the cadherin or whatever it is, could also be something else, right? Is essentially biasing how strong feedback is, right? And we're saying it's kind of different on the dorsal side than from the ventral side, right? And what that is going to do, it's breaking the symmetry along the dorsoventral axis, and that's it. Yes.
I maybe missed this. What sets up the BMP gradient in the chorion?
Yeah, so this is a question from my genetics friends. Yeah, so I mean, yeah. So I think in the Drosophila embryo there's an interesting cascade between dorsal, which is the ventral organizer in the fly, and so then other factors along this axis, which essentially shuttles BMP to something on high levels on the dorsal pole. And there's a young man over there who has a spectacular paper in the making. I hope I'm not giving too much, which kind of looks at these. Yeah, okay, great. So and this is essentially, showing the time course of how this stuff comes up, and they have a really nice physical model. So he'll actually be able to explain much better than I can, how you set it up. Yeah. But I
set up as the egg is essentially being made.
Correct. Yeah. So not as the egg is being made, but as the zygote is being starting, right? So because the egg is made in some different kind of condition. Yeah. Okay, so we've got to put our money where our mouth is, right? So DV symmetry breaking is really important, right? And so what I show you here now is the same plot as I showed you before, with these even skip mutants being really important, right? But now we are sort of removing dorsoventral patterning in the embryo altogether. So this is a mutant called tolarm9. And this is a German word for amazing. Yeah. And what you have here is there's low SMAD signaling within this, which is a downstream readout of BMP, and you lose equatorial gradients within these things. And what you can see is that they do a little bit of germ band extension, just a little bit, right? So they are moving over here and then they're falling back, right? And when you're looking at the accumulated deformations, you can see how this is very different, right? So this is very, very different. There's a little bit of stuff going on, but then it basically stalls and does nothing anymore. No? And the other one is another embryo which has a really cute name. It's called Spätzle, which is a German noodle dish. Yeah, and you know, I mean, Eric came up with all these amazing names, and I mean, not only Eric but Yanni too. Yanni said toll. Yeah, so that's why it's called that way.
Are we doing germ band extension or is the posterior midgut invaginating?
Wonderful question. Yeah, so I actually don't know yet. Yeah, but I would imagine that Thomas would say the posterior midgut is invaginating. Yeah, but I think it's germ band extension, and I'm going to try to convince you of this. Yeah, so Maha was asking, what's the substance that makes this, right? And I said it's BMP, right? If it's BMP, then I have to be actually able to reroute the germ band, right? Because I just took BMP out of an embryo, right? And so, you know, what Matt Lefevre here is doing right now is he's injecting human recombinant BMP, yeah, because for some other reasons we have human recombinant BMP in the lab and it was easy to get by, so we just injected. And so what you can see here is the injections, and I have to applaud to Matt, right, because I'm an asshole, yeah. So I told him, look, I mean, dude, this has to work really well, right? I mean, I want to see that there's no wounds, nothing, right? And he never did these injections, right? So he had to learn this the hard way, and he established everything by now in the lab to a way that I think we are more or less wound-free, yeah. I think we're wound-free, yeah. And you know, what he can do then is stain these embryos for SMAD signaling, right? And this is a TOLR9 embryo, and he has injected SMAD, yeah. And so you can see that we make new SMAD domains appear within these embryos. And what you can see down here is this embryo that has been injected here on the ventral side. And mind you, the germ band should go dorsally. And what you're seeing now is the germ band essentially moving ventrally, yeah. So the germ band actually moves towards where Matt is injecting these things. Yeah, and I think this is a different level of quality, right? Because what we can do now is we can redirect where morphogenesis goes, right? Rather than mutating it. I mean, we mutated it, yeah, it's okay, but we can actually then make it go into some different way. Yes. This would be awesome if I could show you the myosin already. It's in the makes. We want to make movies of this so that we can show you the myosin. Yeah. What I have done is I've looked at Katerin, yeah, and I'll show you that in a second. Yeah. So, and when I do this, it's not me, it's Matt. Yeah. So I'm just pestering him. Yeah. So basically what you can see here is that timing actually matters. When you supply the BMP is actually critical that you do this at the right time. Yeah? So this 0 is when cellularization is completed, and when you do this short before cellularization, there is no effect. Yeah? You see nothing. But when you do it 40 minutes before, you get actually a pretty striking effect. These cells are all moving now towards the wrong side. Yeah? This is something where cells should normally not go to. And it's not injection, because when we do this DMSO, we can see always this tiny little bit of motion towards the dorsal pole. And now I'm getting closer to your question. Yeah.
I just want to make the comment that that's right about the time the gradient points.
Yes. That's great. I don't know these things, but that's excellent. Yeah, that's really. I mean, I wanted to look at it, so I'm glad to hear that you agree with it. Yeah, wonderful. Okay, so I don't have myosin for you, but I have Katerin for you. Okay, so we'll have to live with that, and that's for reasons that antibody stainings with myosin are notoriously difficult in the Drosophila embryo. Yeah? So you know. You can see how there are patches of SMAD appearing when we do these injections, and there's a huge patches now of Katerin that you are creating within this, right? And there is a patch of SMAD, and there's a patch of Katerin, right? And what I really like is that these patches, you know, kind of mirror each other, right? And you make different patches because, you know, you cannot control these injections so well where you are, right? It's not optogenetics, yeah? But it's also a good feature, yeah? Because it explains some of the variability that we're observing within these embryos, yeah.
And you inject it in 2 different positions on the other end. See what happens.
Yeah. I've actually asked Matt to do this. So and he loves you too now, yeah. So yes, and he will do it, yeah. So it's actually doable. You go in and then you go to another position and another position, and that way you can drive this system into different states. Yeah. But this is all ongoing. Yes, please.
This is a very naive question, but is this sufficient to set up stationary flow?
Yeah, I would think so, because it's an external thing, right? So I put it into the shell, right? And that's the source for this, right? And if that thing now triggers, breaks the symmetries within, my mechanical system, right? Then I've broken the symmetry along that axis, right? And that will be a stationary flow. But still to come is that we show you that this is a stationary flow with data, right? But I think in my mind it's there.
Go ahead. But the geometry is going, like the relationship between the geometry of the egg and where you've injected will be different. So you could envision that actually there's a synergy between where it's, injected in the geometry of the egg. It's actually important. Maybe it isn't the same. It's not obvious.
No, yes, it is important. Yes, it is important. Yeah. So this is this slide here. Yeah. So basically, what you have is how fast does the posterior midgut move forward, right? So this is wild type. Wild type is really, really well controlled, right? And what you can do is you can desiccate the embryo. So you remove the turgor, right? And what happens in the embryo is that the embryo kind of becomes wrinkly kind of tissue, right? And that's what you actually do when you do these injections, right? Because otherwise the embryo explodes, yeah? But what Matt has done now is he's basically just desiccated the wild-type embryo. And what you see is the wild-type embryo becomes slower when you desiccate it, yeah? And I would think the turgor is needed to read out the geometry for real in this embryo, right? And what you see here now is the BMP injections are kind of similar. They're not as well controlled as the wild type, and that's mainly the variation within these injections. This is a very hard experiment in my mind to do. I've never done a single injection, but I think, Akanksha, you've done some, so you will be probably able to tell that it's hard. Yeah? And so what Matt then does is he looks at tolerant line and it's lower in speed, right? So we can conclude that BMP is essentially speeding up this process. It's not only telling you where to go, but it also makes it faster. And what I found really spectacular is that when you desiccate now the tolerant line embryo, the speed becomes even slower, right? And what I think these things are hinting at is that there's truly something within this geometric feedback. Yeah? And what I would like to do now is essentially poke the embryo and dial the pressure in the embryo, right? And then see whether we can control speed and stuff like that through these features.
Over the egg, the egg.
Yeah, we have round eggs, yeah, fat embryos. And the fat embryos are actually gastrulating slower. They also have a vastly different look of the myosin on their surface, yeah? I don't have it here in the slide deck, but if you want to see it, yes, it's there. Yeah, that's fitting.
Yeah, say like desiccating would mean you're just slowing everything down. Can you really interpret? Because it's
possible that you're slowing down, yeah. And that's why I want to take the needle, right? And I want to actually control the pressure for real, right? So you come in from the head side, right? And then you dial it up or down and see whether or not. Yeah? I mean, it's an idea, yeah. But what the picture is that we are ending up getting with this is that, we're extending into this space where we say that we cannot just think about the landscape, from Waddington, right? But we also have to think about the extension into, geometry and mechanics, right? And I think that it looks a little bit like we're close to some saddle point in some of these features, right? And by basically adding in BMP, we slightly change the position along the gene expression axis, essentially, if you like, or signaling would be better here, right? And what we do is we're basically channeling the development to now make, germ band extension occur in the other direction, right? And so what that picture would then say is that this morphogenesis is essentially an instability, yeah? And what the embryo is doing is it's basically breaking the symmetry to get this instability in the right kind of way. And now I'm going back to your comment, Lisa, that the shape of the egg is symmetry broken, yeah? And that could be another factor, how I can add, right? And so these things can synergize, yeah? So you can have the genetic signaling and the shape of the egg, together kind of making this process darn robust, essentially. That's the picture.
Could you think about how to frustrate it by trying to inject BMP in a way such that it tries to do the opposite? Like, you know what I mean?
Yeah,
Could you try to
I would not do this with injections because the system is not so well controlled. I would use optogenetic BMP, and then that might work. Yeah.
That'd be cool.
Yeah.
Just continue along that thought. Are there evolutionary experiments that already exist because you have different egg shapes, therefore you have different.
Yeah. Yeah, I mean, yes, there are evolutionary works. I mean, Pavel Tomanchak, Yuqing Wang have looked at this. They looked mainly at the cephalic furrow's presence at the presence of the cephalic furrow
No, no, but I'm saying moving away from Drosophila, going to other systems.
Yes, I mean, yes, absolutely. And I think this is what we are doing right now. We're bound towards this. So we're going actually to a zebrafish now, right? And we're seeing many similar things, that happen in the fly, which is the hydrogen atom. And, you can think of the fish now as the helium or so, right? And it's fundamentally the same quantum mechanics that unfolds within these critters, right? And it's a very different starting point.
I guess I was just wondering about insect eggs.
Yeah.
And insect eggs come in a variety of shapes,
Yeah.
and therefore they also have presumably very different boundary conditions.
Absolutely, totally. And if you give me insect eggs, I will image them.
So I'm thinking also of hemi and holometabolous.
Yeah, I mean, so beetles, could be interesting, I think, right? They also are interesting from the point of view that they do this short germ rather than this long, right? So that could be interesting. Absolutely, totally. Would be really exciting to go into this branch of.
You said that if you get beetles, you'll be able to do it.
Sure. No, I mean, I'll start something, with that young gentleman over there. Yeah, so we'll do something. Yeah. Okay, and so what this says is that, the morphogenesis of a species is essentially just a line, within this manifold, right? And the manifold is actually, some sort of an attractor manifold, but it's a dynamic manifold. Yeah? And so this manifold slowly changes, right? And this slow change is opening up valleys for where the marble being the embryo, rolls, right? And the shape of this manifold is essentially what tells us how mechanics, differentiation, and genetics are actually linking to each other, right? And so I think it would be really, really interesting to measure, what happens outside of an embryo, right? And understand what is going on there. But I've taken already too much of your time, so I won't go into, the thing that happens within, mechanical patterning within an organoid. But if you guys have any questions about this, I'd be happy to chat. I'm still around for some time. So I'm going to breeze through this and get towards the end, and want to just, thank all the people that have been involved in this. Jesus, there's a lot of clicking. And I mean, I guess I can say this here, in this system over here, what we have shown is that you can, or what we hope to show, we haven't shown this yet, what we hope to show is that you can actually have patterning happening with mechanics, and that you can actually pattern muscles in ways, that they are sort of tangentially patterned when they're in this red color, and then when they're in the blue, they're radially patterned. These are genetically the same cells, but they are arranging in different ways, and we can control this. And so what this kind of suggests is that there is also in this branch of mechanics and geometry stuff that happens, more or less independent of the genetics, and allows you to structure this. And so we have this rich phase space, and mapping out this phase space would be interesting. And then I would like to thank all the people that have been invaluable to work with, so all the collaborators and all the great young people who went, all their way now. And the only thing I want to sort of, my funding people, that's great to have those, it's wonderful to get their support. And I want to point out this event that we have, in Santa Barbara. So we actually have, I think, multiple people who are involved in setting up this course here. So what we do is we do a summer school on quantitative biology, where we try to essentially incubate biologists, physicists, engineers to learn, how to do quantitative things with critters, right? So movies, and measure one variable, measure another variable, relate those variables, and learn something about morphogenesis. And Adrian and I are organizing this, and Vanessa and Akangshi have kindly agreed to bring their samples to, the course and toy around a little bit with it. And then thank you all for your attention and good questions.